This video will focus on basic concepts related to single gene inheritance. Some of the main topics we'll focus on are listed on the screen. So for example, we will take a look at this idea of particulate inheritance. We'll also look at Mendel's first law. We'll discuss the basic concepts and stages of mitosis and meiosis, although we will not dwell on these since you likely have had these during some other introductory biology course. We'll discuss briefly the idea of sex-linked inheritance, and we'll also take a look at pedigree analysis in an attempt to understand different modes of inheritance. Let's begin with this notion of particulate inheritance. Please don't feel the need to memorize the text you see on the screen, but the basic idea behind particulate inheritance is that there is something, some entity, some chemical that serves as the genetic material that gets passed on from parents to offspring. And this basic idea was first put forward by Gregor Mendel. Now, of course, we understand and know that the chemical, of course, is DNA, and the particles, if you will, of inheritance are, of course, genes. And we'll talk more about genes later this semester. Mendel was very fortunate. He did lots of experiments on pea plants, and he chose to focus on traits that were either or traits. And he also focused on traits that bred true. So in other words, this idea of true breeding, simply referring to the idea that certain phenotypes or physical traits are reliably passed on generation after generation. So some of the traits he looked at are seen up on the screen here. And he was also fortunate to choose pea plants to work with because he could do controlled pollinations, as we see illustrated here, where he could transfer pollen from anthers to stigmas fairly easily. But also, if you look closely, for example, at either of the flowers, you'll notice that they are bisexual. So pea plants are capable of self-fertilization. So he could not only do controlled cross-pollinations, but he could do self-pollinations as well. So for example, transferring the pollen from the anther of one flower to the stigma of the same flower. So he could do these controlled pollinations and keep track of the physical traits that showed up in future generations. And one of his crosses are illustrated here. For example, he took a look when he looked at crosses between purple flowered plants and white flowered plants. When he did crosses like that, he would transfer pollen to the female part. The female part of the flower, the carpel, would then grow and develop into a fruit with the seeds or peas inside. He could then plant those seeds and let those grow into mature plants and take a look at their flowers. And interestingly enough, in the F1 generation, all of the flowers were purple. So somehow or another, the white phenotype had disappeared. But he could take that one step farther, and he could self-pollinate these F1 purple flowered plants. And when he did that, again, he could self-pollinate, let the female portion develop into peas or seeds, plant those, and in the F2 generation, he not only saw that the white phenotype reappeared, but they tended to occur in a very distinct ratio, namely 3 purple to 1 white. Didn't matter how many 100 or 1,000 offspring he looked at, they all tended to follow this 3-to-1 ratio. He could take that even farther and self some of those F2 individuals. And when he selfed the white flowered plants, interestingly, offspring were always white flowered. Some of the purple flowered plants, when he selfed, the offspring were always purple, but other purples, when he selfed, the offspring were purple or white, and again in this characteristic 3-to-1 ratio. So he recognized that there was definitely a pattern here. There definitely was something going on that sort of controlled this particular phenotype, namely whether or not the flowers were purple or white. So if you think about Mendel's observations and his experiments, you should be familiar with these terms, and you have probably heard these before. But of course, phenotype just referring to physical traits or characteristics of an organism. Genes, we'll talk more about genes later, and they're actually quite complex. But namely, the idea that gene is, of course, a stretch of DNA that typically codes for some proteins. Alleles can be different versions of genes. You could have a gene for flower color. The gene could be either the purple allele or the white allele. Genotype, of course, just refers to the genetic makeup of the cells or the individual that is responsible for the particular phenotype in question. So you have probably at some point done Punnet squares, and certainly you will practice these during class to some extent. I'm not going to dwell on those here, again, because you have probably worked on these before. But you should, for example, be able to do a simple Punnet square for a monohybrid and later on a dihybrid cross as well. But a simple Punnet square for the monohybrid cross we've been discussing for the past few minutes illustrated on the screen here. So for example, in the far left, we can see the phenotypes of the parental F1 and F2 generations. We can take a look at genotypes of those individuals. So purple flowers, let's say, are homozygous dominant in this case. White flowers, homozygous recessive. The slash mark off, you might see that between the letters. That simply indicates that those alleles are on separate chromosomes. But again, crossing a homozygous dominant with a homozygous recessive, each of those produce gametes with those two alleles, either a dominant or recessive. The F1 generation, they're all heterozygotes. They're all purple. You can then do a Punnet square illustrating the cross between selfed F1 individuals. So you simply put the gametes along each side of a square. And again, if you're crossing a heterozygote with a heterozygote, you'll get something like this. And you bring those alleles together. And again, what you see is that in this Punnet square, if we form 4 individuals, this is just predictions here, of course, 1 of those would be homozygous dominant, 2 would be heterozygotes, 1 would be heterozygous recessive. In other words, if you look at the phenotypes, you would have 3 purple to 1 white. Again, this is just sort of a prediction and illustrates possible combinations of alleles or chromosomes in offspring. And again, this matches the 3-to-1 ratio that Mendel was seeing. So he did all these experiments, made all these observations, and Mendel came up with his first law, the law of segregation. Again, don't feel the need to memorize the text you see, but just think about this in your own words. The basic idea that genes come in pairs, right? Think about diploid cells in your body, for example. Pea plants, they're diploid as well. So genes come in pairs that separate or segregate from each other during the formation of gametes. In other words, during the process of meiosis. Okay, so that's the basic idea behind Mendel's first law. Think about the cell cycle for a minute. This, again, should be review. So when cells are dividing, they pass through these various stages, G1, S, and G2. S is where DNA synthesis or replication takes place. But collectively, G1, S, and G2, that's all part of interphase. And again, if this is a cell that's going to divide, it then undergoes mitosis, typically followed by cytokinesis. And so again, you should be familiar with the stages of mitosis. I won't dwell on those here, but think about stages like prophase, metaphase, anaphase, and telophase. And recall again, during mitosis, we end up with genetically identical cells, identical to each other and identical to the cell that we started with. Just think about sort of the chromosome situation of cells undergoing mitosis. When a cell passes through the S phase of the cell cycle, all of its chromosomes form exact copies of themselves. So what we see in the upper left here is an image of a chromosome, a drawing of course, consisting of 2 chromatids. So one on the left and one on the right. Again, theoretically, those are genetically identical to each other. One was formed after the S phase of the cell cycle. Recall that chromatids are joined at the centromere. Kinetochore is just a small region of the centromere where proteins are found that help pull these chromatids apart during anaphase of mitosis. Spindle fibers we see illustrated toward the bottom, they're simply microtubules that again play an important role in pulling those chromatids apart during anaphase. Also think about cells that are able to undergo meiosis. So there's meiosis 1 and meiosis 2. Again, I'm sure you have seen meiosis before in some previous class, so I'm not going to dwell on it here, but certainly you should be familiar with meiosis because really the stages of meiosis serves as the fundamental basis of basic Mendelian genetics. So again, think about meiosis, think about chromosomes separating, forming haploid cells, each of which has half the number of chromosomes of the cell that you started with. And again, this sort of meiosis serves as the basis of Mendel's first law, or the law of segregation. One thing I'll quickly mention about meiosis, you think about prophase 1, you think about the process of crossing over, where homologous chromosomes come together and exchange large pieces of DNA, well, there's this assemblage of proteins referred to as the synaptonemal complex. And again, don't feel the need to memorize the text that you see, but the synaptonemal complex, again, is basically these proteins that hold homologous chromosomes together during prophase 1, and they play a role in crossing over. So again, that's introducing some genetic variation into populations of sexually reproducing organisms. Mendel was fortunate in that he chose phenotypes that were due to genes essentially found on autosomes. But I just want to take a few seconds here and briefly discuss sex chromosomes and sex-linked inheritance. So if you think about humans, we have sex chromosomes where you can be XY, you're a male, XX, you're a female. And so we can talk about genes that are found on those sex chromosomes. So we can talk about either X-linked or Y-linked. Now there are more X-linked genes simply because the X chromosome is larger than the Y chromosome. It does hold more genes than a Y chromosome. One example is of a gene on the Y chromosome is the SRY region, the sex-determining region of the Y chromosome. This is a gene responsible for conferring maleness. So this is a gene that, when expressed during early development, again, triggers the development of a male instead of a female. So what's interesting, if you take a look at genes on sex chromosomes, they display sort of unusual patterns of inheritance. So to illustrate that, we can think about pedigree analysis. We'll take a look at some pedigrees in just a few seconds. But for now, we can think about 4 basic types of inheritance. So some genes we can refer to as being autosomal recessive. For example, the gene responsible for cystic fibrosis, or I should say the mutated gene responsible for CF. Autosomal dominant, Huntington's disease, X-linked recessive, for example, color blindness, or X-linked dominant, for example, one type of rickets. So let's take a look at pedigree analysis and see how we can deduce the types of inheritance based on pedigrees. So some symbols commonly used in pedigree analysis you see illustrated on the screen here. I won't just dwell on these too much, but I'll quickly mention that individuals with the disease or whatever it is we're discussing question, often the affected individuals are indicated by solid colors as we see toward the right. So for example, focus on the pedigree for a few seconds. We could say, well, this is an example of an autosomal recessive. How do we know it's recessive? Well, if you take a look at, for example, this individual right here, or her sibling, her brother over here. So these are 2 individuals that have the disease or whatever it is we're discussing here, but their parents do not. So the parents are probably carriers. They might be both heterozygotes, for example. But the point is, we know this is a recessive allele because these individuals have the disease, but the parents do not. We could say, well, it's probably autosomal. It's hard to tell on a very small sample like we see, but probably autosomal because males and females are affected equally. In another situation, like we see here, this particular disease seems to be showing up in every single generation. So we could say, well, it's probably due to a dominant allele. We could also say, well, it's probably autosomal as well, again, because loosely it seems to be affecting males and females equally. In contrast, if you take a look at this pedigree, again, we could say it's due to a recessive allele, again, because here's some individuals that are affected, and yet their parents are not. If it were a dominant allele, you would expect to see at least one of the parents affected with the disease or phenotype in question, but we don't. We can also say this seems to follow an X-linked pattern because the disease is more common in males than in females. And so the basic idea, if you think about the genotype of a male, again, they're XY, so if they have a mutation on their X chromosome, they simply don't have another X chromosome to compensate for that. Whereas females, having 2 X chromosomes, even if they have a mutation on one of their X chromosomes, there's a good chance that their other X chromosome, it contains the normal allele, and therefore it compensates for the mutated allele on their other chromosome. So again, this pattern that we see here follows the X-linked recessive pattern of inheritance. So that concludes our brief review of single gene inheritance.